Now, our first presenter this morning is Dr. Karla Gage. She's Associate Professor of Weed Science and Plant Biology in the Schools of Agricultural Sciences and Biological Sciences at Southern Illinois University, Carbondale. She completed her bachelor and master's degree in biology at the University of Memphis and a PhD in plant biology with a focus on weed ecology at SIUC. Dr. Gage joined the facility at SIUC in 2015 and enjoys teaching and research in applied management of agricultural systems. Dr. Gage's research focuses on weed control in midwestern U.S. cropping systems and the rotational crops of corn, soybean, wheat, sorghum, and industrial hemp. She has 22 published papers, 3 book chapters, and has authored and co-authored 69 conference proceedings. Please join me now in welcoming Dr. Karla Gage.
So thanks so much for that introduction, Stephanie. It's a pleasure to be here to talk to you today, to give you an update on some of the things we've been doing lately. I also have some resources here for you if you want to pick them up. If you need more posters for your wall, I have a weed management planner, so what you do before planting. I have a summary of the herbicide modes of action, if that information is a little rusty. And then I also have a new take action chart with the products and how they're classified. So feel free to come pick these things up. I'll leave them at the front here. So I want to talk to you a little bit today about, I should turn it on. Okay, there. Okay, so I'm going to give you an update on some of the resistances that we're finding, and then I'm going to talk to you about management tools. So I'm going to start with putting everything into an integrated weed management framework, and then we're going to talk about the value of a pre, what rainfall events do to pre-efficacies, and then I'll tell you about one of the large-scale projects that we're doing on cover crops and planting green. And then I'll end talking just a little bit about some of the new technology and some of the things that we're seeing come out. So to talk about a resistance update, I'll give you an overview, and then I'll talk about water hemp and giant ragweed. I show this graph every single time I present, so you've probably seen this before. This is the increase in unique herbicide-resistant cases globally. This is all systems. It's not just agricultural systems, but there are 522 unique cases with 269 species right now. We know that weeds have evolved resistances to 21 of the 31 known herbicide sites of action and to 165 different herbicides, and this curve is not leveling off. This just shows you the amount of pressure that we're putting on herbicides to accomplish all the weed management work. The more pressure we put on the herbicides, the more we're going to see these resistance cases rise, really. So we've got to diversify our programs, and that's going to mean more than just diversifying sites of action that you put into the field. And these are the resistances in Illinois right now. So Palmer amaranth has 3 documented resistances. Water hemp, of course, we know is our main driver weed here. And you can see all the resistances listed there, but we also have common ragweed, giant ragweed, common lamb's quarters, mare's tail, kochia, giant foxtail, eastern black nightshade, shattercane, and cocklebur that we have to manage resistances for. So we've looked specifically at a lot at water hemp recently. We're involved in another large-scale study funded by the United Soybean Board where we go out and collect escapes in fields. We're looking for specific patterns, and you should do this too. So when you see that clump of plants in the middle of the field, everything else is clean, but you see a clump. It's like a discrete dispersion, right? So you're looking for that pattern, and that tells you that there was a mother plant that escaped control, likely due to resistance, and those seeds are now spreading out from that mother plant. So that's your first indication that there's a problem. And you should also look in the areas where equipment enters the field, so the entries to your field. And that's where you're going to find a lot of the seed moves in, and you may have resistances moving in with it. So we're looking for these patterns when we're looking for escapes. So we're picking suspect resistant populations, and we're sending these out to our other university collaborators for a screen that is just a discriminating dose. They spray these suspected resistant populations with a half rate and a full rate, and then they look at the survivors, and they give us back the data. And then we have been taking those accessions, those seeds, and we've been running what we call dose response curves, where we expose them to at least 6 doses of herbicide, and we try to accurately measure the level of resistance in that population. So this is what we've done. We collect from September through October. We put these in a greenhouse. We spray them with a number of different doses of 2,4-D, dicamba, and glufosinate. So those are the posts that we're targeting. And then we collect the biomass, dry it, and weigh it, and develop our curves. So what we've found is that this is for 2,4-D. We have one population in particular out of all the ones that we've screened that is incredibly problematic. And you can see these are ED50s. This is the y-axis on that graph is the effective dose that kills 50% of the population. The dashed line is the 1x rate of 2,4-D. So you can see population 7 in particular is a resistant population. And on the right, you see photos of that population at different doses, and you can see that it has survived up to a 4x rate of 2,4-D. So we've also looked at dicamba, and we're not seeing, so what we thought were suspect populations for dicamba, they are not, at least so far, the ones that we're screening, and we continue to screen these populations. So as of now, no documented resistances in those populations to dicamba, but we're seeing some issues with glufosinate. And you'll notice it's population 7 again and population 1. Their effective doses are exceeding that 1x rate. On the right, you see photos of population 1, and we do have survivors at the 4x rate of glufosinate. So within the same population, population 7, we have multiple resistance to 2,4-D and glufosinate. So that's work that's ongoing. So the ED50 values, they follow the visual ratings that we're seeing. So everything, our data looks, the ED50s match the visual ratings. So we are continuing, we're ongoing to screen these populations for more active ingredients. Those populations that we confirm resistance in, we will take those and we will characterize them, meaning that we'll spray them with other active ingredients that could be suspect or could be possibly linked to those resistances. For example, 2,4-D resistance has been linked to the resistance of HPPD inhibiting herbicides in the literature. So we will take that 2,4-D resistant population and we will screen it for HPPD active ingredients to see if it's also resistant to those. And that brings me to metabolic resistance. So that linkage, because those plants are able to break down 2,4-D, they have the mechanism to also break down HPPDs. That's metabolic resistance. That's something that other people are working out, but we know that we're seeing more metabolic resistances. And this is just resistance mechanisms in Illinois. Target site resistances, those are mutations in an enzyme that cause the herbicide to not bind as well to that enzyme and reduce its effectiveness. Target site or non-target site resistances, those metabolic resistances, the plant just breaks down the herbicide. It tags it and then targets it and breaks down that molecule, and it's no longer active. So we have, as you can see, a list of those resistances that have been documented. And just to remind you, this is how we get crop selectivity. So crops do this, especially corn. Corn's very good about breaking down the herbicides that are labeled to be applied on corn. This is for atrazine. You can see corn within 6 hours metabolizes 96% of the atrazine it's exposed to. Oats, on the other hand, metabolizes 2%, and that's why oats are so sensitive. So this is something that plants do, and it's something that weeds are evolving to be able to do the more that we expose them to herbicides. And I'm telling you to use multiple effective sites of action, and that is still the best recommendation that we have. You put multiple active ingredients in the tank to control your problematic weeds, and that can also put selection pressure on plants. So the more you expose them to, you may be pushing them more towards that metabolic resistance where they're able to break down multiple active ingredients. So it's still the best recommendation we have for long-term sustainability of cropping systems. But we also need to do other things, right, so that we don't rely quite as much on herbicides, and that's where integrated weed management comes in. I'll talk to you more about that. We also have been documenting this population of giant ragweed. We have a population that is, we already know we have resistance to the ALS inhibitor, site of action 2 in Illinois. That's been present for a very long time. Classic and Pursuit are the documented products with resistances. But we have not documented glyphosate resistance yet in Illinois. We know that it's present. It's present in all the states around us. It just simply has not been documented. So we're going through the process of running the dose response curves again to actually get this documented on weedscience.org, which is the website that most people go to look for resistance information. So we have giant ragweed that can survive up to a 2x rate of glyphosate, at least the populations that we're looking at. And then we're also seeing problems with fomesafen. So the PPO inhibitors, fomesafen has never been just great on giant ragweed, but it has been, 85 to 90% control, or even a little more, has been documented for our susceptible populations, and now we're seeing for this particular population a shift over time. So it is really, it's selection for resistance that's happening gradually, and we're in the process of running more PPO inhibiting herbicides, again expanding this screen to understand fully what we're dealing with. We know that the University of Wisconsin has documented PPO inhibitor resistance in giant ragweed. That's happened relatively recently, and working with Pat Tranel at the University of Illinois, we believe that we have a different mechanism of resistance than what the University of Wisconsin has found. So 2 independent evolutionary events to evolution to PPO inhibiting herbicides in giant ragweed. So that brings me to integrated weed management, right? I've told you we're putting so much pressure on our herbicides. We have to diversify. Diversifying site of action groups is probably not going to be enough. We have to do other things. So the role of integrated weed management is to work with chemical control to increase the different types of control practices that you put into your program. If this is done effectively, it can slow the evolution of herbicide resistance and prolong the efficacy of our biotechnological traits and our herbicide active ingredients. So think of it as I don't know if you know the farm manager, Ronnie Krause at Belleville, who was the farm manager for many years. He would talk about this as thinning the herd. So you reduce the number of plants that are exposed to the herbicides that you're applying through different practices, and that reduces the probability that you're going to select for herbicide resistance. So integrated weed management is the combination of all these tactics to protect our technologies and protect the efficacy of our weed control systems. So we combine chemical control with other management tactics. For mechanical, we could think of that as harvest weed seed control practices. Chemical, of course, we diversify herbicides, and we have our genetically modified crop systems that we work with. For cultural, we could look at diversified rotations, and that's where our lab's research with hemp is coming in to try to diversify our crop rotations and see if there's another commodity that we can put in rotation. But you could also use narrow row spacing, cover crops, of course, which I'll talk about today, and then interseeded crops. And biological controls, we don't have a lot of those available to us, but the crickets that you see in your field and those little black beetles, the carabid ground beetles that you see in the field, they can eat a lot of weed seeds. If their populations are high enough, they could potentially eat 85% of the amaranthus seeds that are in your field. So there are other things that we can do and rely on. And then, of course, this is all on a platform of prevention. We want to prevent those seeds from getting there in the first place, if possible. So now the value of a pre. I'm really proud to be part of this project. So this was led by Chris Landau at the USDA ARS out of the University of Illinois, working with Marty Williams. So I counted 26 authors on this publication, and the title of it is "The Silver Bullet That Wasn't: Rapid Agronomic Weed Adaptations to Glyphosate in North America." Basically, what we did was we took all of our historical data from all of our research programs and we gave it to Chris. And he compiled it and has been running the data to look for trends over all this historical data. It's a very powerful data set that he can now analyze. So we have 11 locations, an incredible amount of data that spans from 1976 to 2022. So we have corn and soybean trials treated with at least one glyphosate application, and then he's been looking at percent weed control. So for now, he hasn't really done a lot with the data, but this one thing that he's done that's impressive, he's looked at 2 subsets. So he's looked at trials with glyphosate only, and then trials that included a near planting followed by glyphosate. And he selected the rates that he wanted to look at. And for each weed species by location, he looked at the efficacy over time, so our ratings of the effectiveness, and he looked at the variability of that control over time. So what he found, you see control on the y-axis there, the percent control by year. So over time with glyphosate alone, we're seeing the control efficacy decrease. That's not big news, right? You would expect that, of course, with the evolution of resistance. But when we add a followed by glyphosate, we don't see the percent control decrease. We actually see the combined effects are increasing. Some of the individual sites are staying the same over time. So we're seeing preserve the effectiveness of that post-emergence chemistry. And the same thing with the variability. So the variability of control has been much greater with glyphosate alone, meaning that some of those populations probably evolved resistance, some relatively quickly. The control was much more variable with glyphosate alone. But when you add a the variability in that control actually decreases. So you get more predictable efficacy. And of course, this is just with glyphosate, and everyone knows that, glyphosate, while it still does have utility, pretty great utility, we have resistance problems that do not allow us to look at glyphosate alone anymore. So we saw that the control control decreased and variability increased over time when glyphosate was used alone. Providing or adding a to the tank can preserve the efficacy and reduce the variability in glyphosate, but this can be applied to all of our other chemistries as well. So pre's are important to preserve the efficacy of what we're doing here. And just as an aside, he's also looking at the effect of weather on our post-herbicide efficacies, which, again, it's such a powerful data set. He here has looked at, on the y-axis, the average temperature 10 days before application, and then the total rainfall you see along the x-axis there. And then the colors that you see are the probability of successful control, 1 being full control, and 0 or the yellow being no control. So as temperature, with low temperatures and an increase in rainfall, we see water hemp efficacy dramatically decrease. And then same rainfall after application, we see with higher temperatures and higher amounts of rainfall, we have dramatic decreases in efficacy of water hemp. So these are the types of things he's going to be looking at as he mines the data. So just in summary, increased rainfall and higher temperature after application led to reduced control. We will see climate shifts. So as global temperatures continue to rise and rainfall becomes more variable, our ability to consistently control these and other common and troublesome weeds is at risk. So we should understand these relationship patterns. Even though we can't control the weather, right, we can at least predict what the outcome of our management actions may be. So now looking at extreme rainfall events, I just wanted to share with you a project that we just completed. And again, this goes back to the importance of soil residual herbicides. So those pre's are key management tools to fight herbicide-resistant weeds. We have weeds with extended emergence patterns. Palmer amaranth and water hemp both have extended emergences. I wanted to put this graph back up. This is old data from 2015, actually, looking at the density over time, or the seed density per meter square of Palmer amaranth over time from May 12th to beyond September 29th there. But we have these different treatments. Two of those that you see close together are tillage treatments, and the red on top there is no-till. Our average pre-application timing occurs when there's already some emergence of Palmer amaranth. So there'd already be some plants emerged in the field, possibly at the time that you're applying that pre. And then, of course, at the time that our post is applied, we still have that emergence curve that's continuing after that post-emergence application. So if you don't put in a residual with your post, you're going to have continued emergence of Palmer amaranth and water hemp at these times. Now, I also wanted to relate this back to the practice that we're seeing of people shifting their planting date of soybeans earlier and earlier, right? So the earlier you plant, the less time in the season you're going to get on your pre-emergence herbicide. So what are you going to do 21 days after your pre-application when it's no longer effective? You need to come in with a post. Hopefully you're putting in another soil residual in that post, but you still have this emergence pattern. So are you going to go back with a second post application to maintain your weed control? And there are a lot of people that are working on this now to see how weed control fits into this early planting strategy. So we have been looking at the efficacy of group 15 herbicides following severe rainfall events. So what happens when you don't get enough rain, and then what happens when you get too much rain, and what is too much rain? What are the limits here? So we know that the climate change models are predicting higher rainfall in the spring in Illinois, followed by more extreme dry events. We know that the group 15s rely on incorporation, usually rainfall, to be efficacious. And group 15s are essential components. So there are overlapping soil residuals that are applied at that post-application timing. So we wanted to look at the limits of rainfall on the group 15s. We did this at the Agronomy Research Center in Carbondale, and we used S-metolachlor, which is dual magnum. We used acetochlor, which is encapsulated in acetochlor, which is warrant, dimethenamide P, Outlook, and pyroxasulfone or Zidua. And then we simulated rainfall events in these different quantities, 16 centimeters, 8 centimeters, 4 centimeters, 2 centimeters, or 1 centimeter. And we did this with irrigation. So we laid irrigation pipes. We split the field into 5 irrigation application timings, and then we sprayed and then followed it with irrigation with these different rainfall amounts. So we had these application timings within a 7-day period. We did this in July when we were not getting ambient rainfall. So all of our moisture that we applied in the field came from our irrigation timings. And this is just an example of what we found. So this is our visual control ratings for common water hemp again. So you can just see for our 4 different pre-emergent herbicides that we looked at, we had dramatically different efficacies over these different rainfall amounts. Now, the top curve here, that doesn't show up. The black dashed line is encapsulated acetochlor or warrant. So it really didn't lose that much efficacy until beyond 8 centimeters. So the encapsulation for water hemp actually held its efficacy over time. The red dashed line is Zidua, the green line is dual 2 magnum, and then the most sensitive one was Outlook or dimethenamide P. Now, I tell you this to suggest that, I know we can't control the rainfall amounts. We have no idea what the rainfall amounts are going to be when we go into the season, but it might be good to understand the interaction of the chemical that you're using with rainfall. So how sensitive is that chemical to degradation by rainfall? How easily is it going to be washed into the soil profile and deactivated? And then that'll kind of tell you more about what your timing should be as far as your next control measure. So when we look at the summary here, common water hemp, rated 28 days after application, a rainfall event of 4 centimeters was enough to reduce the efficacy of S-metolachlor. 16 centimeters reduced acetochlor, 2 centimeters reduced dimethenamide P, and 8 centimeters reduced pyroxasulfone. When we looked at the count data, so we counted the number of water hemp plants in each plot, we saw that acetochlor and pyroxasulfone showed enhanced control compared to esmetolochlor and dimethenamide P, but rainfall events over 4 centimeters caused a decrease in efficacy. So what we found was that the effect of rainfall was highly dependent on the chemical that you use. Acetochlor was better on common water hemp than on tall fescue. I didn't show you the data for tall fescue, but dimethenamide P was better on tall fescue than water hemp with those rainfall amounts. So it's going to vary by species, of course. And then because we saw the greatest level of control on tall fescue from acetochlor at rainfall events between 4 and 8 centimeters, we presume that the encapsulation may have negatively impacted the ability of the herbicide to perform at low rainfall events. So it took more rainfall to see that efficacy on tall fescue. So in the future, we would like to do a direct comparison on the encapsulated versus non-encapsulated formulation of this product, which I think would kind of tease that out. And then overall, the data suggests that pyroxasulfone and acetochlor are more stable under high precipitation events than esmetolochlor and dimethenamide P. So again, just another thing that's important to consider as you choose your soil-applied herbicide. So now I want to tell you a little bit about a study that we were involved in 2 very large-scale studies looking at planting green. So the interaction of cover crops and termination timing and herbicides. This is one that I haven't presented yet. So this is a new. I'm sharing this with you for the first time. So when I say planting green, just for audiences that don't understand what that is, you're just planting soybeans into a standing, living cover crop biomass. And then you're terminating that after planting at some point, a day or more after planting. So this is a project that's led out of Penn State. So John Wallace is the primary investigator on this, and this is his student, Grant Hoffer's project. So I'm showing you data that Grant has assembled. This is a presentation that he's just recently given at a regional conference. The title was Delayed Cereal Rye Termination Influences Weed Recruitment, a Regional Perspective. So there's a lot of talk about planting green. It's definitely gaining interest because it can increase the biomass that's present in the field, which has soil health implications, as well as, of course, weed control implications. So the more biomass you have in the field for your cover crop, the better your weed suppression is going to be. So working through GROW, which is a network called it's called Getting Rid of Weeds. So that's the acronym there. So it's all about outreach, and this is just one of the projects that GROW has set up. It's a huge group of scientists with lots of different projects. So we're looking to evaluate the effects of delayed cover crop termination on weed and crop management across a soil and environmental gradient. And you can see in blue the states that are involved there. So we want to inform these regional decision support tools. So we want to gather all the information so that we can better make decisions about how to manage cover crops in this planting green scenario. That specifically is looking at increases in cereal rye biomass production. So how do we increase biomass production? Broadleaf weed recruitment patterns at our post-application timing. So what does that biomass do to broadleafs at post? We're looking at the efficacy of soil-applied residual herbicides. So are the soil residuals working with that amount of biomass in the field? And then, of course, soybean yield. So we believe, or the hypothesis is, that planting green will increase weed suppression alone and in combination with soil-applied herbicides, but the magnitude of this effect will vary based on the gains in cereal rye biomass that occur across production regions and driver weed species within production regions. So we at each of our sites, we set up a high biomass treatment and then a lower biomass treatment. So I have 2 fields each year of this study where I'm trying to get different levels of cereal rye biomass within our sites at Carbondale. Our main plots are cover crop, either no cover crop, which is the control, CTL. We have cereal rye with a standard termination, 14 to 21 days pre-plant. So this is the 14 days pre-plant, DPP. And then we have the third one, cereal rye, planting green, terminated 1 to 3 days post-plant, or 1 day after plant, 1 DAP. And then we have these split plots where we look at either a post only, we don't apply a pre, or we have a 2-pass program with a pre followed by a post. And each of those main plot treatments, those cover crop treatments, are split with those herbicide treatments. And then we used Enlist 1, and our pre was Fierce XLT. So we collected cover crop biomass prior to termination. We looked at the weed density by species, collected 28 days after planting in 2 meter quadrats, 2 meter square quadrats. And soybean yield data was collected. We then did a weed density analysis. Grant did a weed density analysis using the relative response index. So I won't really go through that equation really, but I will show you the data. And this is what we're doing. So this is the framework of the entire project here. So we're looking at cover crop management strategies, specifically cover crop residue mass, and then we have our cover crop management legacies, our soil percent, volumetric water content. We're looking at soil temperature effects, and then soil nitrogen pool effects. And nitrogen pool goes back to how we're trying to drive differences in biomass. So we have a high nitrogen field that should get high cereal rye biomass, and a low nitrogen field to drive the lower biomass that we're looking at. And then we're looking at the weed management legacies, so the propagules pressure, the amount of seeds that are emerging, and then species traits. Seed size, of course, is a component here. We expect the smaller seeded broadleafs to be better suppressed by cereal rye biomass than the larger seeded ones. Larger seeded weeds should have more energy, more ability to push through biomass layers, so we're looking for that distinction in our data. So I want to focus on the graphs on the left, and that's just cereal rye biomass at termination, and you can see just the color coding is all the different states. In general, the longer the termination window, the more cereal rye biomass you get. That is mostly in the high biomass treatment, so where we had higher nitrogen in our fields at planting, we see higher cereal rye biomass. When we have lower nitrogen in planting, the termination window does not matter as much across more sites. So we get less cereal rye biomass whether or not we terminate before or closer to planting. And then we looked at the response of broadleaf weed species. So this is just probably difficult for you to see, relative responses of annual broadleaf weed species. So weed density at 28 days after planting. So we saw that weed suppression increased in our 14-day pre-plant termination timing in the control at 7 out of 16 sites. So even in our control treatment, we still got increased weed suppression at 7 out of 16 sites. 14 DPP with the pre, we had weed suppression at 13 out of 16 sites. So more, the pre added efficacy there. And this is, again, termination at 14 days before planting. At 1 day after plant, in the control treatment with no pre, we had suppression at 8 out of the 16 sites. And then when we added the pre, we had suppression at 14 out of the 16 sites. So there wasn't a ton of difference in this comparison, whether we terminated at 14 days prior to plant or 1 day after plant, as far as weed suppression goes. We did not see significant yield differences either among those treatments. There were 2 locations that showed patterns where yields were decreased in Nebraska, a drier climate, or increased in southern Illinois when planting green relative to the control. This is a little different than our other large-scale project that we've looked at, where you can see soybean stand counts on the y-axis by cereal rye biomass. We see a decrease in soybean stand counts, and then on the graph on the right, you see soybean yield by cereal rye biomass, and we see a decrease in soybean yield on average, with higher cereal rye biomass. Now, there is a cluster of points, right? So this is a regression curve, and all the points in the lower right of that graph are pulling it down. You see a good number of clustered points up there where you have higher cereal rye biomass, and it is not affecting soybean yield. But we know that the possibility is there. The more cover crop biomass you have, potentially you can see some impacts on yield. We saw it in one of our large-scale studies, and we did not see it in the other. So now that brings me to cultural and technological practices. Just a hint, at current and future trends. We're seeing much greater adoption of harvest weed seed control practices, and this is probably not a great option for everyone. But if you're having trouble with some fields where you have high levels of herbicide resistance, you really don't have a lot of other chemical tools left, this is an option. So if you do this over time, you're destroying the seeds that go back into the seed bank. And for these summer annual species, if you do not let them produce seeds, then you've effectively won the battle, we could say. So this is a relatively new approach to tackle herbicide-resistant weeds. A lot of this technology is coming out of Australia. Australians have a very similar challenge when it comes to herbicide resistance, so there's been a lot of innovation. So the I guess, trick to this would be, to make this work, the weeds have to retain their seeds at harvest. We see a lot of seed shatter as the combine moves through the field. As soon as the header contacts that weed, seeds can shatter, and if they don't go into the combine, they can't be destroyed by these practices. So the weeds have to retain their seeds. We would predict over time, if we start relying on this type of technology, that we're going to see weeds that evolve to shatter early. They're going to mature earlier, they're going to reproduce earlier, and they're going to shatter easier. But for now, we see very high levels of efficacy for the amaranthus species. So if you have multiple resistant water hemp in your field, this is potentially an option. You can destroy the seeds before they go back to harvest or back to the field. There are a number of different practices, six different ones in particular, that have come out of Australia. Some of them involve collecting the seeds and the chaff from the crop and taking them off-site. That would be the chaff cart, where you go dump that somewhere or you feed it to livestock. Narrow windrow burning is something we've looked at as well, where you funnel not only the chaff, the entire crop residue is funneled out the back of the combine in a huge line, and then you set it on fire and you destroy the seeds. And that can be effective as well, but of course then you have issues with open fire, with burning in the fields, and of course, especially in Australia in such a dry climate, that is probably not ideal. And then there's a bale direct system where the residue is just baled and taken off site. We have chaff lining, which is something that my lab is working with quite a bit. It's one of the lower hanging fruits of harvest weed seed control. All you have to do is build a chute and fix it to the back of your combine. And that separates, you have to have some separation within your combine. So if you have an older combine model, this might be more challenging. But you line the chaff, so the most fine particulates of the crop out the back of the combine, and that's where the weed seeds go as well. The straw is spread back on the field. So you put the weed seeds in a line, in the same line, every year, every time you harvest. Over time, you can clear the surrounding area of 94% of the seeds. And this has been shown to happen within 3 to 4 years. You can isolate the seeds of the amaranthus species in this line. Now, it's not going to look pretty, right? Because that's where all your amaranthus are going to come up the next season. But you can go back and you can band applications on this line. And there's some pretty great work right now looking at the interaction of the chaff lines and soil residual herbicides in corn following soybeans. And you really get a longer window of control when you combine those two practices again, chemical and chaff lining. Impact mills, again, these are now being built into combines. They destroy the seeds before they go back onto the field. They devitalize them by crushing the seed coat. And then chaff tram lining, it's the same thing as chaff lining, but you put the chaff lines out the back of the combine in the tire tracks so that you run over them every time you move in the field. So another form of control. Chaff lining in particular, though, that's something that since it's a very, very low cost, I think that could be a realistic management strategy where we could ban herbicide applications on the chaff line and improve control. And I just wanted to, this is almost the end here, I know I'm running out of time. This is, again, integrated weed management. So herbicides alone, and then herbicides plus harvest weed seed control. This work is from Michael Walsh, who's one of the leaders in harvest weed seed control technology out of Australia at the University of Sydney. So you see in the top line there, well, we have on the y-axis annual ryegrass plants per meter square, and then along the x-axis we have the year of these treatments. We see much greater efficacy when we combine the practices and use herbicides and harvest weed seed control together. The top line is herbicides only. So the bottom line is herbicides plus harvest weed seed control, and you can see that many fewer plants, annual ryegrass plants, per meter square, and then by 2010 and 2011, you've effectively reduced that population to almost zero by using those practices together. So we're also working in the digital space. My lab is supplying images to, again, the GROW network, and they're building image detection systems that are open source that any company will be able to access and any grower will be able to use. So we're building this image repository of all the weeds that we have within our collaborating states across the nation. And then we're providing this data set that will be available to train software programs. So if you're a startup company trying to get into this space, you don't have to spend tens or hundreds of thousands of dollars obtaining the images. You have an image database to start with that you can work with. So we're seeing a lot of movement with weed detection and control. I mean, John Deere See and Spray is a great example of that, but there are so many others. So this is technology that we're going to continue to see improve. I have a colleague that's working on this. He's actually set up his own consulting company, and he estimates that there are over 250 different companies that are working in this space right now to develop autonomous solutions for weed control. So we're going to see a lot of movement in the technological sector, which will, again, relieve some of the pressure on our herbicides, but it's all about using these things in combination. We can't just switch from one tool completely and adopt another. We have to make sure that we keep as many tools as possible available for our use, and we need to try to diversify and move beyond just a chemical focus. So with that, I thank you for your time. It was really great to be here today to talk to you all. I don't think I have any time for questions, do I? Okay.
Are there any questions?
Yes.
On your rye, when you're using a cut crop, you drill it in, and you broadcast it after harvest, and what was your seeding rate?
That's a great question. So we have worked with anywhere from 60 pounds an acre to 110 pounds, and you don't really see, I mean, over 100 pounds, you're not seeing any increase in weed suppression. The going rate right now for most of our studies is around 60 pounds, so that lower rate. And I think that's pretty much the standard in most of the weed suppression publications that I'm looking at. So I do believe that was the seeding rate for all the data that I showed you. We do drill it. We don't work with broadcasting it, right? Because that will reduce the establishment. So yeah, we've been drilling what we've been using. If we did broadcast, I would want to up that rate for sure. Yes.
Hello.
Hi.
Yeah, I just wanted to jump in because I had a question also related to the cereal rye. So, and maybe you mentioned this, but what would be the ideal amount of biomass that you should shoot for? I mean, if there's too much biomass, then it reduces the soybean yield. How much do you really need to get the benefits for the weed control?
Yeah, that's a great idea, a great question. I can't remember the ideal biomass amounts right now. I know that there are a number of publications that address that, and there are regression lines that have been developed to try to look at that relationship, and I really can't give you an exact amount, but yeah.
Are you looking at the weed suppression just in the bean part of the rotation with the cover crops, or in a corn bean rotation?
Most of our work has been in soybeans only. Yeah, so we don't, partly because crop rotational studies are difficult to fund, right? A lot of our funding comes from the soybean board, and we really try to focus on soybeans in those projects. And then the funding for long-term projects that allow assessment of rotations is just limited. Most of the funding that my lab has a 3-year duration, and then I should say reinstatement of those projects for continued funding is never guaranteed. So rotational studies are hard for funding, I guess is what I'm trying to say, and we've really focused on soybeans.
And to pair with that, is there any look or follow-up about the influence of planting cover crops after the herbicide programs that you implement in those soybean studies?
Yes, so there has been talk of that. Is a demo trial to look at some of the most important herbicide programs in both corn and soybeans. These are our showcase trials, and they're always placed at the Belleville Research Center. So if you ever want to see what the going trends are and what each major company is promoting as far as their portfolios, you can pop into Belleville and take a look at what we're doing there. And there's always somebody there to give you a tour. They can take you out to the field and show you the list of our treatments. We want to plant cover crops after those showcase trials and start collecting the data. There have been discussions about how we can do this. I think that would be a really important data set to have. We have not been able to put together the resources to make that happen yet for many different reasons. We plant those trials late so that they can be shown at our field day, which is mid-July. So we can't plant at the normal planting time. We have to push that back a little bit so there'll be something to see. And then that means our harvest is later, which means we can't plant our cover crops in a timely manner unless we change the way that we do things there. But that is something that I really want to see, because that would give us a really great look for all the major programs that people are recommending. How is that affecting our different species of cover crops that we're trying to plant? So yeah, that's something we want to do but haven't. Okay, thank you all.